On the streets, my brother's dying, and I hear the mothers crying, crying out, and the way that things are going.
So I'm here to talk about the histopathological analyses and how to actually then evaluate what the GM industry has done and to see whether or not it's adequate. So I investigate things using a microscope. So Judy's been talking about what you would use without any assistance, where you just splay open a stomach. I actually then take the samples and I look under a microscope. So now, as she had said, I'll be looking at something else. So firstly, I wanted to go through two particular things. Just some vocabulary. So genetic code. We heard Don Hoover mention that when it comes to DNA, it's found in a nucleus and it's actually coiled together. And then when it's being read, it uncoils and then it's read. So genetic code is like a recipe book. You've got several little different recipes, and each of those things that the cell produces starts with what is called a promoter, which is the start. It tells that this is where the recipe starts, and then it reads the full recipe, what you need for it, how much of it you need to produce whatever it is by the cell, and then it's got a terminator gene. So that's genetic code. It has the steps and ingredients. Secondly, histopathological analyses. It's what I do. I'm a histologist. I work at the University of Adelaide, as you saw on there. And histopathology is the study of the structure of organs under a microscope. And then pathology is the study of disease. So histopathology is the study of the tissues and if there is a disease. Now, as Judy had mentioned, firstly I wanted to mention the Bt toxins, because I think some of the farmers use them in organic farming, and the differences between the GM crops and the non-GM crops. So there's what is used in organic farming in the sprays is a bacterial spray. You spray the crop and the bacteria produces these spores, and the spores have this crystal on the very end. But that crystal is not automatically insecticidal. It needs certain steps made for it to actually be insecticidal. So this is what happens. So the crop is sprayed, and then the caterpillar comes along and actually eats the leaf that has been sprayed, and while it's been sprayed, these bacteria produce the spores which have the crystal. And so while the caterpillar is eating the leaf, it ends up eating the spores. And the crystal has this capsule. And the capsule dissolves away in the digestive tract of the caterpillar. And it needs a non-acidic environment. A non-acidic, am I correct? Yes. A non-acidic, sorry, it's very late at night in Australia, so we would be in bed by now, but yeah. So it needs a non-acidic environment to dissolve away. So us humans, we have an acidic environment, so keep that in mind. It needs a non-acidic environment to dissolve away, releasing this thing which is called a prototoxin. Now that is an inactive toxin. Still not active, it still cannot kill any bugs. The enzymes in the digestive tract of the caterpillars then cut that inactive part, so that white part there, and release a toxin. Now that toxin then binds to the wall of the gut, and that then induces a cascade of events which then kills the cell and then thus killing the caterpillar. Okay, so these particular events are very important. So you need the crystal capsule to be dissolved, the non-active part to be cut away, and then it needs to bind to the actual wall itself. So the difference between animals and humans and caterpillars is the fact that we have an acidic stomach, so we cannot dissolve away that crystal. We don't have the enzymes to cut because they are specific for the caterpillar, and also we do not have the receptors on the wall. for that toxin to then be toxic to us. So that's why it's deemed safe by the organic farmers in production. Now, what happens in BT toxins? Well, quickly stating, there's no crystal capsule. There's hardly any bit of a non-active part, and the receptor binding may not be required. So, what we have here is the genetic code. So remember, that's the recipe book. So we've got a start here and a stop there, and it will read the active toxin, and then it will read the non-active toxin, and then it will produce it and release it by the cells. So the industry found that when they put the full recipe in, it had little to no effect at all on the insects. So when they included that whole genome into the whole recipe, into the GM crops, it had hardly any effect on the insects, on the caterpillars at all. So what they did, they removed majority of that structural part. This is all quite sensitive. They removed majority of that, and they found that it had nearly about 100% success rate. So they've actually removed one section of it, so there's no capsule anymore, because it's just the BT toxin, and they've removed that part that might need to be cleaved as well to activate it. Now also, what they've also done is they've actually increased it, they've changed the genome even more to increase its insecticidal properties. So all of a sudden the difference is quite great. It's quite different between organic farming and the BT toxins found in GM crops. So we have no crystal capsule, possibly hardly any of the non-active part. Receptor binding may not be required. Now they've found that the insects are actually becoming more resistant to the BT toxin in the GM crops. So they are actually now producing GM crops without that non-active part. They've completely removed it. Actually, no, without the receptor binding part, so not the non-active part. And then the genetic code has been changed to increase the toxicity. So clearly, this is a completely different product. Now let's look at the genome itself that is put into a GM crop. Now this is Roundup Ready soy. And I'm sure a lot of us know what Roundup Ready soy is. It's a soybean that when you spray Roundup, it will be able to withstand its action. And it withstands its action through the use of EPSPS. But the interesting thing about this is that EPSPS isn't taken out of nature completely in full. As you can see, it's got two parts to it, but let's just step back a bit. As I said, we've got a promoter here, and we've got a terminator. So the promoter says, start reading, and then we go along and we start reading the EPSPS from a petunia, which is a flower. And then we start reading it from an agrobacterium, which is an actual bacterium that produces tumours in plants. And then it will go and continue on to a terminator, and it will say stop. Now let's look at another GM crop. This particular one, I want you to notice that the Cry1AB, which is a BT toxin, it has been synthetically developed. So it's completely developed in a laboratory. It hasn't been taken out from nature. It has been completely made up. The whole recipe has been made up in a laboratory. And then also there's no terminator present in this one. It was lost while they were producing the GM crop. So quite precise methods, isn't it? The other thing about this particular maize is that when they were producing this GM maize, they put in 2 recipes into that maize, but only one stuck. The other one got lost somewhere. So it's when the GM industry likes to say, we know what we're doing, it's very precise, it's better than crossbreeding because we know where it's going and where it's happening to it. Well, not quite right. Okay. So let's get into what I actually do. So Judy covered a little bit about the different safety analyses. And in a rat feeding trial, you've got different models of safety evaluations of feeding trials. The important thing is to remember is that a rat's life is 2 years. It's why it's a good model to use, because you can actually feed it for 2 years and see the whole the whole development of disease. So you've got different models of studying pathology or pathology development. You've got acute, you've got subchronic, chronic feeding trials. What the industry most often does is this one, just the subchronic, which is 3 months, which is 1/8 of the rat's life. That's all they find. In something like cancer studies, from a histopathological point of view, cancer does not show up until 12 months. So if the industry is doing three-month trials, is it a thorough study that they're actually doing? So this is what Judy and myself were really fighting for, that the industry, when they do a feeding trial, fine, you can do a 3-month trial, but then your methods have to be more specific, more thorough, and just using quite sensitive methods. This protocol I got from a laboratory, which does not want to be named because of repercussions that may occur, but they study piglets, and this has been revamped a little bit as well for rat feeding trials. So they use a similar one for rat feeding trials as piglets. With the piglets, they want to know how early you can take a piglet off a mother's milk and give it normal feed so it can go to the market. So that's the reason why they do their feeding trials. And if you think about the GM industry and what Judy was saying, a lot of the feeding trials are on pigs and seeing whether or not there's as much meat being produced in pigs or chickens and whatever. These guys are doing roughly the same. They want to make sure that these pigs are ready for market. But this is their protocol. They do the feeding trial, then they kill the animal, they collect the tissue, and this is for the digestive tract, that's my field, and in particular this is the small intestine, and they will stain sections, they will dye the sections using 2 different stains, and I will show you why that is. So, but you've got an H&E and you've got an ABPAS. The H&E is what the industry will use, and they'll only use that. Then they look at the measurements. They'll do all sorts of measurements to see the lining, whether or not it has thickened or lessened, and you can tell then if it's healthy. If anything has lessened, then it might be erosion, there might be something happening there. Then they'll do cell counts. They'll actually look at how many, is there a loss of cells, because the cells will tell you the environment of the gut as well. So if you've got an increase in goblet cells which produce mucus, it will mean that the acidity has increased, so you need to counteract that acidity. So they'll actually do. Then they'll do this immunohistochemistry, which is very specific. They will look for cell division, which is looking at the product of Ki67, and they'll look at apoptosis, which is forced cell death. And then this particular one at the very end is quite novel, and it's only been around maybe for the last 10 years, and I think this one is the most important in the short-term feeding trials, because that one, if you've got cancer starting, it will start in the nucleus of the cell. And so if you stain for this, then you know that the cell has mutated. It might end up becoming cancer one day. So you can do a 3-month feeding trial, but then look for these specific changes. And how will they look? Well, this is actually the apoptosis. This is cell dying, but this is a tumour. And they've this is mine actually. I've specifically labelled it for the cells dying. And so the blue are actually the nuclei of cells that are not dying, and the brown are the cells that are dying. So all of these brown dots are dying cells in a tumour, in a rat mammary gland tumour. So you would do exactly the same thing for the mutation of the DNA, and then you would get blue and brown. Very simple, positive, easy to do, well, roughly easy to do, but it's highly specific. So let's look at what the industry does. Now, this particular study was done, has just been published, and it was in response to Seralini's 2-year rat 2-year study looking at cancer, where he actually found the tumours, and you saw that in his speech. So they wanted to go back and redo that study. But there are a lot of problems with this study. In particular, there's this issue of blinding. Now, Judy mentioned it slightly before. When you're doing a study and you give the tissue to somebody, if they are pro-GM, if they want to have pro-GM, well then they may not want to find anything. Now, if they're anti-GM, well then maybe they want to really find everything and they will just look for it too much. So that's called a bias. So when you give a researcher tissues, and Judy does that to me, she gives me the tissues and I don't know at all which ones are from each group. So I have no bias. I cannot, I cannot even subconsciously, accidentally start looking for things or not see things. Well, they've decided not to do that because it will be too costly and too much time, because what they want to do is just to look at the samples that are from the GM fed. That's it. That's what the industry usually does. When they decide not to do a blinded study, they only look at samples from a GM fed study. If they find something in the GM fed, then they look at the control and they look only for what they found in the GM fed group. And if they find that same change in the non-GM fed group, then they say it was not caused by the GM crop. So they really cut corners here. They don't look at all the samples. And also, a very important point is this is a novel food. It has never been in our food supply. Our animals have never eaten it. We have never eaten it. So the model of damage may not even be known. So they're actually looking for models of damage of which they know about. They are looking for diagnostic models already, because that's what they're looking for. They don't actually measure anything. They don't do any cell counts. They look for just superficial things. If there is a definite cancer there, then that's all they'll look for. But if you do the measurements, the cell counts, then you know what is really happening to the tissue, and you may end up finding that GM crops cause something completely different. So, yes, they're saying that they're not doing it blindly because they may not notice subtle differences. I think we've covered that already. If you're using things like the blue and the brown, you will see subtle differences. You don't need to unblind your study to be able to do that. And significant decrease in comparability. If you've got an exact same protocol for your GM and your non-GM, then that automatically makes them comparable. Does it not? I don't know. This is logic for me. If you're using exactly the same methods and treating all your tissues exactly the same way, that makes them comparable. But for some reason they seem to think that won't make them comparable. I don't get that one. Now, I wanted to say that this proper studies have been done in the past where they do, roughly proper studies, where they do actually do mucosal measurements. Now this is in Russian, so you may have to take my word for it. But this is what it says. You've got your mucosal measurements, so the thickness of the walls, and then you've got your cell counts, and that was 2009. Now this paper has actually been used by Monsanto as well for their safety analyses whenever they're applying for approval. They will actually, for Roundup, is it Roundup Ready? For that particular corn, that particular maize, they will actually use this one as evidence. And the same thing goes for this soybean. And they've also got a 30 days and 180 days. So it's possible. Clearly it was once upon a time done, but not quite. Anyhow, I'm wondering how we're going with time, but this is now our study that we did with Judy. Now we decided to do a subchronic feeding trial. We wouldn't be able to find any cancers, and we weren't actually looking for cancers because that's another thing. And I'm planning to actually do the nuclear stain at some stage to see whether or not our changes. But the mucosal measurements and the cell counts are already give you quite a bit of information to look at. But this is This is the regular stain, so the H&E stain. This is the first part of the wall of a stomach. So you've got mucus-producing cells here, and then you've got the glands, and you've got a muscle layer here, and then you've got other layers. So your food will be up here. You can see some of the mucus up on this end. And this is what the industry will look at, something like this. And they'll say, oh yeah, it's roughly normal. And then they'll look at another one and they'll say, yeah, that's roughly looks normal. They won't measure. They won't measure to see whether or not this is thicker than that one. However, as I said, the protocol asks for another stain, and this is ABPAS, and all of a sudden, the pits become easy to see. So this is the mucus-producing area. If it's increased, it means that the acidity, so this is stomach, the acidity has increased in the stomach. So something in the food is really causing problems. It's reflux or who knows what. So that actual thickness will end up changing. Neck region. Oh, this is very sensitive. The neck region here is where new cells are produced, and they will then mature and migrate upwards, or mature and migrate downwards. So this is the proliferative zone. If in cancers, for example, the proliferation is increased greatly, and so you often want to know if this area is proliferating highly. Also, if you've had erosion or any damage, then this area will be activated, and there will be a lot more cells in this area because new cells have to be produced to take up the space of those that have been lost. So it's a lot easier to see here, but with proliferation I would have used that blue and brown stuff. So let's now go back to this, and some may have noticed this little structure here. Now normally that structure is normal in a rat that is aged. So the older you get, the more problems you've got, and the slower your body acts, and the same thing with rats. So this may actually happen, but remember our feeding trial was only 6 months, which is 1/4 of the rat's age, and that's not even old as well. But the industry told us, oh, this is normal because it's found in aged rats. Well, yes, ours are young, so of course it's not normal. But we did find this structure in both groups. So according to the GM industry's protocol, if you find it in both groups, that's it. It can't be caused by GM crops. But because we did a secondary stain, so this is all these glands again, they're called dilated glands. Because we did the secondary stain, we found something different. This is blue. I don't know if you can see, it's blue. Well, this one has a more purpley colour. It shouldn't be blue. So there is epithelial dysplasia, it's called. So the cells have changed properties. So my next role would be to figure out what properties have been changed. Is it a mutation? Could it be continuing on to become cancer? Things like that. And I'm running out of time, am I? Sorry. The other thing that we found was these glands had cells that were all elongated. They were stretched. There were 2 models that could be happening. One is that there had been a cell lost in there, and so the others had to stretch out to cover that space lost. Or the gland has actually expanded because there's been an increase of production of something. Anyhow, there are other methods that can be used. Now, how much time do I have? Doesn't matter? Okay. There are other methods that you can... It does? Okay. There are other methods that you can use which are specialised. We've covered immuno. You can also use immuno. But what we did was also electron microscopy. So we took an area, something like this, zoomed it in, and was able to look at individual cells. So this here is the nucleus, and then this here is one cell, and then this here is another cell, and then there's another cell here. And then you can actually look at the individual properties of that cell, the different organelles. This is all mucus up here. And then you can see whether or not the 2 cells are well opposed. So do they keep together? And what we found in the GM fed, now I did this blinded. So I noticed this change, took note of it, and then afterwards I gave my results to Judy, and she was the one that noticed, hold on, this is only found in GM group. So I didn't know, I just simply noticed, actually it was my students. Isn't it wonderful to have students? Mm-hmm. They noticed this change. So if we zoom in, you can clearly see that there's nicely approached. It's like shoelaces. You've got these laces lacing the whole thing up, while here, some of the laces are still connected, while some have been given way. And in this one, there may still be a little bit of the lacing left, but the majority of it. But it doesn't mean that things are going through. Just like in a fence, you have holes in a fence, but depending on what it is, it's not going to be able to go through. Like a fence, a goat cannot go through a fence, but sand will be able to pass through. So depending on the properties, and so you can do further studies to figure out whether or not it's a leaky fence. And I think we can end it there, or do we want to? I just wanted one more thing. This is my very last slide. Okay, this is the industry and their feeding trials. Now their published feeding trials started in 1999. Well, if you look at 43.2, this is Roundup Ready soy. That's their published feeding trial, but it was already approved for human and animal consumption somewhere in the world, a lot earlier. And then we can look at GA21, which is a corn, if I'm not mistaken, or a 1810. It's a corn, yeah, both of them are corn. So GA21, a year earlier, it was already approved and released somewhere in the world for human consumption. And so this line is all the different GM crops already approved, and these are the studies, the feeding trials. So the ones here have never had a feeding trial. The ones here, in between here, have never had a feeding trial published. Isn't that interesting? Anyhow, that's it. Thank you.
Thank you very much.